During the process of DNA replication, we are going to copy both strands of DNA, and we're going to use this using a couple of principles. We're going to use the principle of anti-parallel replication or anti-parallel strands, and then also complementary base pairing. And complementary base pairing is going to tell us what each new daughter strand is going to be based on the original parent template. So in lecture, we would have gone over the mechanism of this. DNA polymerase is the main enzyme that is going to replicate both of our strands. And remember that during DNA replication, we are making DNA out of DNA. So we're going to maintain all of the same nucleotides. Remember that A always pairs with T and C always pairs with G. Again, if we know one, we know the opposite strand. And what we should end up with is two identical daughter strands that are not only identical to one another, but identical to the parent strand. So if we go ahead and get started with our complementary base pairing, on this top strand here, we're going to start at the 5' end, and we are just going to insert our complements using our base pairing rules. And so on all the way down our sequence. And so if we continue to do this, we end up with two strands that are identical to one another and also identical to that parent strand. So I would like you to take a minute. Feel free to write down the parent strand here, practice your replication, make sure that you're maintaining DNA to DNA, which means we keep all of our 4 base pairs. Once we understand the process of DNA replication, we can use this to further our understanding of molecular biology by looking at what happens during the process of gene expression. Gene expression happens in 2 steps. The first step is transcription, where we are going to use one of our DNA strands to code for a messenger RNA. And then that messenger RNA is going to be translated into a protein. So our messenger RNA is basically going to be the code that tells us what amino acid we need to put in. So first we need to get the information out of our DNA molecule, and we are always going to use the template strand. The way that this is written at the moment, our top strand is going to be the template strand. I will always tell you which one is the template strand or give you enough information for you to figure out. But there are 2 things to keep in mind here. One is that we always only use one strand. So we're going to copy one strand, and we always copy the template strand. And I'll tell you why we copy that in a second once we see how we copy it and we see the resulting sequence. The other thing that you need to remember is that you are going from DNA to messenger RNA. We are transcribing, we are writing down, and we are going from nucleic acid language to nucleic acid language. But this is like going from British English to American English. The language is the same, but there are some words that are different. And in this case, the word, in air quotes, the word that is different is uracil. And instead of A binding with T, instead A is going to bind with U. So if we see an A in our template strand, we need to insert U as the complement. But T in the template still binds with U in the complement. So if we see a T, we are going to insert an A. Nothing changes with C and G. We don't have to worry about those. They're still going to bind the same. So let's go ahead and write out our sequence here. Again, anti-parallel rules still matter. So we are going to start from the 5' end. T complement is A. A's complement is U, C's complement is G. AAA reads U, U, U. TGA reads A, C, U. So notice every time I have an A, I put in U for the complement. If I have T, A is still the complement. So don't get those two mixed up. Now, the other thing that students get mixed up is they want to make a copy of both strands. But if we think about the end result, we are using the top strand here to make a messenger RNA and ultimately to make an amino acid sequence. This messenger RNA here is a very specific sequence. If we use any different sequence, we get a different amino acid sequence for our protein. So if we look at the second strand here on the bottom, the 5' strand, it is a different sequence, which means we are going to get a different protein. Our cells use only one strand at a time, and one strand is used to make a copy of the DNA into messenger RNA, and that messenger RNA is used to make a protein. So we're only going to copy that one strand. I would like you to take a minute, use that template strand to practice making your messenger RNA. Make sure that you put in the correct U's and A's wherever they need to go. I mentioned the template strand, and here I told you that the top strand was the template strand. The other strand is called the coding strand. Now, the reason we call it the coding strand is that this one that we're not using to make our messenger RNA is actually almost identical to the code that we use to make our protein. The only difference is now we are in the language of RNA, so we're using uracil. So our template is the strand that we use to make the RNA version of our coding strand. That's why we call them template and coding. But we are always going to use that template strand. So our next step is taking this mRNA sequence that we have and translating it into a protein. So in translation, we are going to go from that messenger RNA to an amino acid sequence. And what we're doing here is translating our language from nucleic acid to the language of protein. So we're making a completely different language here. So this is like, instead of going from British English to American English, it's like going from American English to a completely different type of language, like Chinese, where we have a completely different character set, different language rules, different grammar rules, etc. So in order to translate this sequence, we are going to use our codon chart. This codon chart is pretty small, so I would suggest that you open up your lab manual or use the codon chart that I have in class to help you determine your sequence. But we are going to start from this 5' end, and we are going to find AUG. So AUG is down here on the bottom. AUG codes for an amino acid called methionine, but this methionine is a special one that only happens at the beginning. If we see a methionine later, it's going to be a regular amino acid. This methionine has a special little tag on it that says it's the first amino acid in the sequence. Then we're going to find UUU. So UUU is phenylalanine. So that's the second amino acid in line. Then we're going to find ACU. That stands for threonine. And we are just going to continue to do this. I would like you to pause and continue to do this as we go. You can practice. But the end result should be this list of amino acids. So there are a couple of things I want to point out in this sequence. One, we've got methionine at the beginning, and we've also got a methionine in the middle. Again, the one at the beginning is a special one called F-methionine. It's just a special version of methionine that only happens at the beginning. The one in the middle is just normal methionine. It does not mean that we started over. It doesn't signal the beginning again. Only the first one signals the beginning. And then our very last one, UAG, codes for one of 3 stop codons. The stop codon is basically a place marker. There are no amino acids that are put there. It's essentially an empty transfer RNA that sort of jams things up. We don't bring any new amino acids. We bring in an empty transfer RNA, and it signals the ribosome to release all of these parts, release the messenger RNA, release the brand new amino acid chain. In lecture, we will talk about the mechanism of this, but for now, we're just going through what happens with the sequence. So again, we are going to start with our template strand. We are going to use our template, that's our template there, to make our messenger RNA. And then we are going to use our messenger RNA to code using our triplet code and our codon chart to decipher that code and get the sequence of amino acids. You will also have one more handout that is an optional handout in most semesters. You need to double check what semester you're in, but most semesters it is just an option just to practice these gene expression steps. Now I like this one because it shows the shapes a little bit. It shows you that DNA is double-stranded, mRNA is single-stranded, but it also brings in transfer RNA. So transfer RNA is an RNA molecule that is going to be attached to an amino acid. And its job is to read the mRNA strand and bring in the appropriately coded amino acid. So transfer RNA also could be thought of as translator RNA. It is going to read the messenger RNA sequence and convert it into the amino acid sequence. And this all happens inside the ribosome. So our instructions say we're going to fill out the DNA strand. You know how to do that at this point, so go ahead and take a minute, work through the first one. You can just check it. I'm going to fill in the sequence, and then you can come back. We are working on DNA, so you need to be using thymine. And then we are going to use, in this one, it says use the bottom strand as your DNA template. So this one, we're going to use the bottom strand. And we are going to use that to produce our messenger RNA. So we're going to take our sequence here, make the complement. But remember, we are making RNA, so we need to put in a U anytime we find an A. So we are going to start AUG. GUA, and I'm going to go ahead and just fill them in here. I started to put in the wrong letter there, so make sure you catch yourself. Usually if students make that mistake, they get over to their codon chart and they realize they've put in the wrong one very, very quickly because there are no T's on that chart. The problem comes in when students have to decide if they're doing replication or transcription, and then you have to decide if you're putting in U's or T's. All right, so transfer RNA has the anticodon or the complement to the mRNA. So here we are going to use the mRNA as our template to make the transfer RNA. Now, what's happening here is that the mRNA is both coding for the amino acid. That is the sequence that we have here. We have the mRNA codon here in our chart. But in order for the tRNA to bring in the correct amino acid, it has to recognize the messenger RNA sequence. And it does that by having the complementary base pair. So we are going to make the complement again. Again, we are RNA, so anywhere we have an A, the complement is U. If we have a U, the complement is A. And then we are going to use our codon chart to determine the mRNA codon for our amino acid. So we are going to start up on the messenger RNA again. AUG is that special methionine. GUA, looking at our second triplet, is valine. GCU is alanine. AAC is aspartame. No, I messed up the name on that one. Asparagine. Sorry, I said the wrong one. And then last one is CUU, which is leucine. You do not need to have all of the names memorized. I have most of them memorized just from doing this for so long. You also do not have to have the triplet codes memorized. You are always going to be given a chart. So there are 3 or 4 more gene expression pathways that you can go through on this handout. I highly suggest that you do it. Usually students get it after a time or two. Make sure you're paying attention to what sequence you need to use as the template and which sequence you need to use for the complement.